Meiosis Project by Valerie Yunus and Benjamin Milred. All cells undergo something called the cell cycle. The cell cycle is a series of events from the time a cell divides to the time its daughter cells divide again. Next is reproduction. There are two types of reproduction: asexual, consisting of mitosis, which makes clone cells, and then sexual reproduction, consisting of meiosis, and the offspring from meiosis are different than the parents. Cells. Somatic cells, also known as body cells, are diploid, and they are in mitosis. Next are gametes, otherwise known as sex cells. They are haploid, and they are in meiosis. Diploid cells. Homologous chromosomes in pairs. They have the same banding pattern, size, shape, and centromeres in the same position. Chromosomes. There are two different types. Autosomes, which are non-gender specific traits, for example, eyes and hair. Next are allosomes, which determine gender. It's two X's for woman, X and a Y for a male. These are gender-specific chromosomes. They are in things like eggs and sperm. Inside of humans, we have 22 pairs of autosomes and 1 pair of sex chromosomes. So in total, that is 23 pairs of chromosomes. Meiosis. In meiosis, there are 2 stages. Meiosis 1 performs reduction division and reduces chromosome numbers, going from 2n2x to 1n2x. Then there is meiosis 2, where multiplication division is performed, increasing the number of offspring cells, going from 1n2x to 1n1x. This is the cell cycle for gamete precursor cells. It starts off with interphase, which consists of stages G1, S, and G2, with no new DNA synthesis between divisions. Next is meiosis 1, which has a chromosomal number of 2n2x. Then, lastly, it goes into meiosis 2, which the chromosomal number is 1n1x. Interphase. It has 3 stages, the first being G1. In G1, the cell grows, develops, and differentiates. The chromosomal number during this stage is 211x. G0. At the G0 checkpoint, the cell loses its ability to divide. That is why it is given the name all or nothing checkpoint. If it passes, the cells commit to dividing, or if it doesn't pass, it stops growing. The next phase is S phase. In S phase, DNA synthesis occurs, the DNA is copied, the chromosomal number is 2n2x. After that is G2 phase. In G2 phase, the cell gets ready to divide, makes protein, stores food, replicates organelles, and the chromosomal number is 2m2x. After G2 phase is the G2 checkpoint. In this checkpoint, it checks for DNA damage. If there's no DNA damage, it sends it off to meiosis. If there is DNA damage, the cell will attempt to fix the damage. If it cannot, it sends it to apoptosis, or cell-programmed death. Meiosis 1, which is reduction division with a chromosomal number of 2n2x. It starts off with prophase 1, with the same chromosomal number of 2n2x. First, centrosomes, made up of 2 centrioles, duplicate and migrate to opposite poles of the cell. After that, homologous chromosomes pair up in a process called synapsis into tetrads, otherwise known as 4 chromatids. Next, the nuclear membrane disintegrates. After that, non-sister chromatids cross over a crossing over point called the chiasma. The next phase is metaphase 1, with a chromosomal number of 2n2x. In this phase, the centrosomes are at the poles, and chromosomes are lined up in pairs at the metaphase plate. They're connected by non-kinetochore microtubules. The kinetochore microtubules connect to centromeres on both chromosomes. This is called independent orientation. After that is anaphase 1, with a chromosomal number of 4n1x. During this phase, the cell elongates. Non-kinetochore microtubules lengthen, and kinetochore microtubules shorten. The homologous pairs of chromosomes separate, and centromeres do not divide. Then there is telophase 1, where each cell has a chromosomal number of 2n1x. During this phase, a cleavage furrow forms. A new nuclear membrane forms. Chromosomes begin to uncoil. actin microfilament ring begins to form, and the ending chromosomal number is 1n2x. The last phase for meiosis 1 is cytokinesis 1. In this phase, each nucleus is haploid, having a chromosomal number of 1n and 2x. The DNA in the daughter cells are different from the parent cells, and the two cells have been created. Finally, we have meiosis 2. During this process, cells do not undergo the cell cycle between meiosis 1 and meiosis 2. The first phase in meiosis 2 is prophase 2, with a chromosomal number of 1n2x. During this phase, chromosomes are duplicated, the nuclear membrane disintegrates, and a spindle apparatus made of spindle fibers of kinetochore and non-kinetochore microtubules begins to form. The second phase is metaphase 2, with a chromosomal number of 1n and 2x. During this phase, the chromosomes line up on the metaphase plate in single file, and the centrosomes are at the poles. The chromosomes lined up are in pairs and connected by non-kinetochore microtubules. The kinetochore microtubules connect the centromeres on both chromosomes. This is another example of independent orientation. Next is anaphase 2, with a chromosomal number of 1n, 1x. During this phase, the cell elongates. Non-kinetochore microtubules lengthen, kinetochore microtubules shorten, and sister chromatids separate. This causes the centromeres to split and be pulled apart. After that is telophase 2, with a chromosomal number of 1n, 1x. During this phase, a new nuclear membrane forms, chromosomes uncoil, a cleavage furrow forms, and a ring of actin microfilaments starts cytokinesis during late telophase. And finally is cytokinesis 2, with a chromosomal number of 1n, 1x. During this final phase, chromosomes uncoil to form chromatin. Each nucleus created is unique, and 4 cells that are created are different from the parent cell. And this concludes our cell reproduction project. Thank you.